Victoria Sanon Gomez. Victoria, welcome. If you want to share your presentation, Victoria is from Brazil, and she will present us Liponium, an algorithm for identification of disparate traits associated with Mycobacterium TB heteroresistant and virulent.
Good morning, good afternoon. Today I'll talk about Liponium, an algorithm for identification of discrete traits associated with Mycobacterium tuberculosis, heteroresistance, and virus. And here I'm Victoria, and here are the other authors who contributed to this work. Well, to start talking about heteroresistance in tuberculosis, we need to know a few informations about this disease. So, tuberculosis, also known as TB, it's an infectious disease caused by a group of closely related mycobacteriums called Mycobacterium tuberculosis. And it affects about 10 million people worldwide annually, and with causing 1.3 million deaths, which 95% of those deaths were reported by developing countries. And tuberculosis became a huge, important health problem due to increase of multi-drug resistant tuberculosis, and referred as MDR-TB, is the result of inadequate treatment. The MDR-TB is defined as the resistance to, at least, isoniazid and rifampicin, two commonly used drugs for TB treatment. And we also have the extensively drug resistance, XDR. There are the resistance to group A drugs that includes some of the most powerful antibiotics to TB treatment. And those two types of drug resistance are known as heterodrug resistance or just heteroresistance. And there are some phenotypic tests that allow us to find those mutations, identification of the drug resistance mutations, and among all the genome of all the variations of Mycobacterium tuberculosis. But the current tests based on MGIT systems are time-consuming and really complex, so it destimulated its use. We don't want to use that often. Here we can see illustration of how we sequence the TB DNA from the bacteria sample to the whole genome as digital data. So we start extracting the DNA from the bacteria in the sample. Since it's a sample gotten from a human, there could be any variations of those bacteria, not all the same. So we need to extract of all of them. After that, the DNA must be fragmented. And in the third step, we start the DNA lab preparation. And in the first step, we do the sequence itself using next generation sequencing tools and methods. And however, when we get the results from the sequencing, the DNA is still fragmented, so we need to reconstruct it. And we do that through bioinformatics tools that do this task. This way we get to our 50th and last step, ending with the whole genome reconstructed. Well, bioinformatics tools are great allies for the information extraction problem, and they receive raw next generation sequencing reads as input and provide us all type of analysis, speeding up the extraction process. And bioinformatics tools, it must be at least rapid, providing quite quick turnaround time, They must be specific for the problem we want to resolve. They must be also computationally inexpensive, because not only computational research are very expensive, but time is directly proportional to it. So the bigger the computational cost, more time the tool will need to perform its work. And finally, we need the tool to be fully validated, so we can trust its results without the need of double check it. However, and we, despite its advantages, we're still facing some problems with those tools. Um, just a few bioinformatics solutions aren't available for the identification of tuberculosis resistance, but the existing ones are really computationally demanding. And also, the portability of accessible solutions is a challenge, and those solutions require the whole genome assembled before a priori to start its work, which creates an extra step on the analysis process, consequently increasing the computational and time costs for the whole analysis. So, considering what has been talked into here, our objective was to develop a computational, inexpensive, and portable informatics solution for the specific detection of tuberculosis heteroresistance from next generation sequencing reads. And for that, we developed Liponium. Liponium is a Python-based tool that has unpaired Illumina sequences reads as its input through FASTQ files, not being necessary to work with the whole genome, but it can work with it too. To detect these mutations along the sequences, Liponium have a set of custom and resilient probes, allowing target mutation detection, that is, look for drug resistance mutations related to a certain medication, avoiding mismatches during the process, consequently reducing the results unspecificity, like false positives or negative results. Well, talking about Liponium implementation, it is composed by two layers. The first and highest layer is developed in Python, and using libraries like fuzzy search and pandas for nucleotide matching and codon filtering, and BioPython to codon translating into amino acids. The second layer and the hidden one use AWK in bash that is called from the Python layer and are responsible for mapping the probes into the sequences. And for this implementation specifically, we used the AWK version. Here we have a flowchart illustrating the Liponium workflow. And the execution starts mapping the probes into the sequences from the input. And that's the part when the bash layer works. After that, we go to the second step, and that here is when fuzzy search works. And it looks to find identify the mutation codons in the sequences. And it uses the nucleotide referenced and find at the previous step as a way to start verifying if there is, in fact, a mutation or not. It checks the nucleotide and also its neighbors to confirm or not the mutation. And then, in the third step, Liponium will filter the results by its quality, because sequences with a bad quality indicate a possible error in the sequencing process. So the results obtained from them are not reliable. But this way, only the results from good quality sequences will remain. So we can go to our fourth step, because they are more truthful. Here is when the codon translated to amino acids occur using BioPython library. And at this step, Liponium generates a report file with all the mutations found, the information about it, and what every mutation means. And with that, Liponio ends its execution.
Sorry, Victoria,
Here,
for letting you know that you have 1 minute left.
okay. Uh, the materials, a method used for, we use it for the Liponium tests. We used it 274 sequences from minor variants, and the process of the bioinformatic analysis. So we can go to results. Here we have an example of the reports produced by Liponium, and the most important columns that we must talk about is the gen column that has the names of the genes that suffer the mutation, the mutation type, the reference codon, which is the codon of the gene before the mutation, and the mutation codon, that is the new codon after the mutation. We have the count column that indicates how many times this mutation occurred in each sequence. And we have also the reference amino acid, that is the reference codon after the translation, and the mutated amino acid after translating the mutated codon to amino acids. And here we can see more explicitly what the mutation did to our gene. And also we have the comparison between TB Profiler and Pont Finder. There are two well-known tools to identify TB mutations, and Liponium. The yellow circles are Liponium's ones, the blue are Pont Finder's ones, and the red ones are TB Profiler. And we can notice that there's a lot of overlaps, and that represents mutations that all three methods could identify in the sequences used. Here's the name of these genes. But we can also see that Liponio could find a lot of mutations that the other two tools couldn't identify, showing that Liponio can do its work effectively and could get in great results. So, to conclude our presentation, we can conclude that Liponio is a rapid, portable, and inexpensive bioinformatics solutions for the identification of tuberculosis heteroresistance. Our algorithm does not require the whole genome assembly, but reducing significantly the computing time and its complexity. And Liponio currently works, Liponio can infer heteroresistance from raw, unpaired, and paired scaffolds and context sequences reads. And by now, Liponio works with Illumina inputs, but we are working to make Liponio able to work with other next-generation tools, such BackBio, IonTorrent, and Oxford Nanopores. The virulency mark detection is also an important problem. Oops, and will be added in future releases. And here we have the name of other people who helped us to develop Liponium, and I want to thank them all. And that's it. Thank you for attention, and I'm opening to questions.
Thank you so much for your presentation, Victoria. This is a really nice development. We had a question of Diana, and I was also thinking to do the same one, that it is if the software is really available to the community.
We have reposed on GitHub with the Liponium code, so everyone can download it and install on its machine.
Okay, perfect. Thank you so much. And I have a brief question too. Do you know, or do you remember about how many time it could take to process one sample or one genome, for having an idea?
I don't have the exactly numbers because we're working on that kind of tests, but it takes only a few minutes in the worst cases to analyze
okay,
that.
it's really fast.
Yeah.
Okay, thank you so much. Really nice development. Congratulations.
Thank you for the opportunity.
Thank you. So let's move to the second speaker, Daniela Senra. Daniela. Are you here? Yes.
Hello.
Here, Daniela. Hello, Daniela from Argentina. And she will present her work entitled Stemless Score for Single-Cell RNA-seq Data Using PPI Network as a Scaffold.
Hello, everyone. Thank you for this opportunity to share my work. My name is Daniela Sembra. I am a PhD student at CONICET, Argentina. And today I'm going to show you some research we've been doing with my advisors, Nara Isoni and Luis Diambra. First of all, I'm going to introduce you to the type of data we are going to use. So standard methods to like microarrays or RNA-seq, standard methods that measure gene expression provide averages of gene expression levels from large populations of cells. So if you have mixed populations of cells with different cell types or states, we will not appreciate those differences. So this is when single-cell RNA sequencing comes. Cells are isolated, and then what we sequence each cells individually. So what we finally have are expression profiles for each individual cell. So the data we will be analyzing are expression matrices or count matrices. These matrices dimensions are around a few, from a few hundreds of cells to thousands of hundreds of thousands of cells, and around dozens of thousands of genes. Genes are usually located as rows and cells as columns. So the typical applications for this data, there are many, but some of them are obtaining gene expression signatures or uncovering rare cell types. So now we have introduced the data we'll be using. I'm gonna talk to you about what we have done. We developed an algorithm that was called Origins, which was implemented as an R package. The purpose of this algorithm is to quantify the potency states from single-cell RNA sequencing data. Usually, researchers use stemness markers to identify stem cells, but sometimes that information is not available, and we provide a different way of identify stem cells that does not need that prior information, because we already include information from curated databases as protein-protein interaction networks and gene ontology information. So once we identified stem cells, this is useful to set the origin of trajectory inference when performing trajectory inference. So let's go to the method. We used two databases. First, Pathway Commons, where we downloaded around 2 million of interactions, protein-protein interactions. And then on the other hand, we downloaded the proteins associated with the cell differentiation process for humans, and we integrated these data sets. So we kept, as we have is shown here, the interactions that are associated with the cell differentiation process. This protein-protein interaction network ended up having around 200,000 interactions. So once we have built the protein-protein interaction network associated with cell differentiation, we wanted to quantify how active this network is. So for that, we proposed this method where here you can see a graph where these nodes are genes and they are connected with these links or edges. And we propose that these edges are proportional to the gene expression levels of both nodes. So two genes known to interact at the protein level, we have greater interaction probability when they are both highly expressed. So here you can see that once we have calculated these edges, we sum up all these contributions, and we end up having this score, which is called activity. Here A is the adjacency matrix that has ones when there is an interaction and zeros otherwise, and X is the expression level associated with the genes. So to sum up the method, we input expression matrices that have to be normalized and pre-processed. Then we input this into our algorithm, and the output is a vector which size is the number of cells the expression matrix has. So for each individual cell, we have one score that is called activity. There are other methods to quantify the potency state of individual cells. There's a group of methods that are based on statistical mechanical principles, specifically based on entropy. So what these methods are based on is that undifferentiated cells have high entropy levels. This means that they have the possibility to differentiate into different cell types. Undifferentiated cells are associated with heterogeneous expression profiles. So we used an implementation which is called Lancet of these entropy-based algorithms. Another method that is available is C2Trace. Authors found that gene counts are correlated with the state of differentiations. So this algorithm finds the genes that correlate with gene counts and then creates a gene count signature, which is specific for each data set. And they define a score that quantifies this potency. But this has a limitation, especially with quiescent stem cells. This method is not the best for identifying quiescent stem cells. So we tested our method with healthy breast tissue. And I am briefly introducing you to this tissue. We are in particularly interested in the epithelial part of the breast, which is basically composed by luminal epithelial cells and basal neoepithelial cells. Most of the breast tumors arise from these epithelial cells, so that's why we're interested in identifying stem cells and then how the differentiation process is. So we used a data set that is publicly available. It has around 4,000 of cells. We had to filter, eliminate potentially damaged cells, doublets, debris, multiplets. That filtering process is shown here. Then we have to normalize the expression matrix so we can compare cells among them. We perform the typical pipeline. We perform dimensionality reduction techniques such as UMAP and PCA. Then we cluster cells, perform differential expression analysis, and annotated cells. The results are shown here. Here we can see we obtained 3 main clusters, and we perform differential expression analysis, so we obtain the markers for each cluster. we annotated these clusters as base. This pink one was named a basal cluster. This one was called luminal 1 or luminal precursor cluster, and this one were the luminal 2 or luminal mature cells that are associated with the hormone responsive part of the breast. So then we perform all the methods I mentioned previously over this data set. And these are the results. We obtained that for the entropy-based algorithm and for our algorithm, the basal cluster was where the cells that had the highest potency scores. But for cytotrace, that did not happen. And well, that the basal cluster had the highest score is what we expected, because stem cells in the epithelia, in the breast epithelial cells, are located basally. And here is an approximation of a method. It's the same method, but the input matrix is reduced to the highly variable features. So this is a much more faster approximation of our method. Then we benchmark other features of our algorithm. We tested these algorithms with 4 data sets: breast epithelium, colon, hematopoietic, and lungs. All the methods are positively correlated, being the highest correlation coefficient for our method and the entropy-based one. Then we quantify efficiency. The most time-demanding program was the entropy-based. The least demanding program was cytotrace, and ours was in the middle. Now we have diminished our time. Then we benchmark other qualities, such as the memory usage. This is important because the cytotrace and the entropy-based algorithm for the breast dataset, for example, needed around 6 GB extra of memory, while our algorithm needed around 27 kilobytes of memory. Then, well, our method has a simple biological interpretation. And another quality our method has is that it can handle zeros in the expression matrices, which is another important quality, because most expression matrices, it depends on the normalization, but they are sparse matrices and they have lots of zeros. So the entropy-based algorithm can't handle zero-containing matrix, so you have to use other normalization techniques. And well, our method, you only have to type 4 lines of code in R. And well, here is an application of our method. Once we have identified, quantified the potency score, we set the cells with the highest potency score as the root for a trajectory inference. Trajectory inference is basically ordering cells on a time variable. And well, we set the root of these trajectories as the highest, the cell with the highest score. And we obtained 2 main lineages going from the basal cluster to the luminal 1 cluster, and another lineage from the basal cells to the luminal 2 cells. And both of the lineages go through an intermediate state in the luminal 1 cluster. So once we have...
Daniela, only for letting you know that you have 1 minute left.
Yes, I am finishing. Thank you. So once we have performed trajectory inference, we can identify the genes that are correlated with pseudotime for both lineages. So these genes are the ones that drive differentiation. Well, this is what we have done here for both lineages. Here are the genes that are important in this differentiation process. And I just wanted to finish by saying that if you want to read more about this work, our work is publicly available here. All the data used was not ours, and it was publicly available on the GEO database. And the package is also available on GitHub. So that's all. Thank you. And well, I am open to questions.
Thank you so much, Daniela, for your presentation. I think you have a really nice development and very useful tool. And thank you also for sharing the publication and the GitHub repository in which all of us can find your tool. And you are receiving a lot of congratulations to your work. I have a question. Maybe I am not an expert in the area, but I am interested to know, how do you level your clusters? Because you mentioned that you were assigning the levels, but I am not sure how do you do that.
Okay, so this data set was published, and the authors they labeled the clusters, but yes, but it was not the clusters. The annotation was not publicly available, so we used their markers. We performed differential expression analysis, and we used markers, yes, to label the subclusters. Yes.
Okay, thank you so much. And are you, because I saw that your experiments were performed on human data, are you planning to try to use the tools in another species? Of course, that I suppose you depend on being able to find public data sets for doing that. But
currently, we are not planning on. The protein-protein interaction network was specifically made for humans, so we will have to modify the protein-protein interaction network. What we are planning to do, I don't know if it's related, but what we aim to do, and we are doing it right now, is to testing this on breast cancer. We are interested on identifying cancer stem cells.
Okay, thank you. Steffi, do you want to ask a question?
Do we have time?
Yeah.
Short question. First of all, congratulations, because this work was amazing. My question is that about the protein-protein interaction network. So maybe you said, and I didn't get, it is included in the package. So do you have the network in the package as a file?
Yes.
Yeah.
Yes, on our packages, you can include data too. So
yeah.
it's included. Yes.
Perfect. Thank you.
Thank you so much.
Okay.
Thank you, Daniela. Congratulations for your work.
Thank you for this opportunity.
Thank you. So let's move to our third speaker. She's Fabiana Rodriguez de Goes. Fabiana, you're here? Yes, you are. Fabiana is from Brazil, and she will present today her work entitled Feature Extraction Based on Complex Networks for Classification of Genomic Sequences.
Thank you, everyone, for watching my presentation. I'm going to talk about feature extraction based on the complex network for classification of genomic sequence. Okay. This is a project that I'm starting, so I'm going to talk about some aspects of the research, like introduction, DNA sequence classification data sets, data modeling, feature extraction importance, some related works, and at the end, I'm going to talk the project goals. Due to the advancement in sequence techniques, the generation of genomic data has increased significantly in the recent years. So this is a good aspect, because the sequencing and the study of DNA is a fundamental step for the technology and the study of the genome that carries the information about an organism. So in this context, DNA sequence classification is a process that determines if a given sequence belongs to a specific class. So the objective is classify a DNA sequence according different classes. So this is an important problem in biology. But analyze this huge volume of genomic data in order to identify significant partners and informations is a challenge in medicine and genetics. And machine learning has become important too because it enables the automatic extraction of potential informations and about relationship from large and complex data, like genomic data. As well, complex networks and graph theory have been used in different bioinformatics research because biological networks provide a natural representation of complex systems. So complex networks enable the representation of different biological systems and help to study and analyze these systems. So this work presents the summary of a project that aims to investigate and propose new approach based on complex networks and machine learning for DNA sequence classification. So the main objective in this project is to study and investigate approach to model genomic sequence as a graph or a complex networks for partner extractions to be used in a classification task. I'm going to talk about some data sets and some classification tasks like splice junction. So it's possible to classify a DNA sequence according to splice junctions. In this example, we can classify a sequence as 3 types of classes. Another example is classify DNA sequence as promoter regions. So it's possible to classify if the nucleotides is from a promoter or not. And another example is to classify DNA sequence as holding, promoter, or intergenic region. So we have here 3 types of classification and data sets. I'm going to talk now about data modeling. That is an important step in this research. So it's possible to have different data modeling. The first one is a traditional data modeling that is where an object is represented by a vector. For example, a flower. A flower has a color. A color is a characteristic, has a number of petals, and all these characteristics is converted to a vector that represents the flower that is an object. So this is a traditional way to modeling a set of objects. The second one that I want to show is a graph, is a complex network, where it's possible to map the relation between the objects. So in this case, you can have, for example, a graph of friends, and the connected between them represent the friendship. Another example is protein interaction network that map the interactions between the proteins. So you can use this type of graph to extract information, to study a set of objects. The next type is heterogeneous networks that maps the relation between different types of objects, like drugs and genes, patients and genes. These machine learning methods can be applied in different types of modeling. You have graph-based methods applied to graphs or complex networks, and traditional machine learning methods that performs in traditional representation, like feature vectors. One important point is the performance of machine learning methods depends directly on feature representation. So you need to have good feature representations, extract good features to achieve good performance. So the idea is to find an advanced approach to extract features from the data. In this project, the objective is to use complex networks, use graphs as a modeling to be used to feature extractions. So this is the main idea. The idea is that the complex network you provide advanced features that will be used by machine learning methods to perform sequence classification. You have some related work that try to find and propose different advanced feature extraction and modeling, DNA sequence modeling. So have an approach that use convolutional neural networks in sequence modeling as a text data. So the author used text mining to model the data. We have some works that use deep learning models to extract useful features directly from the DNA sequence. So it's interesting works. We have some works that use complex networks to model the sequence, and then these networks are used to generate features. So the connection between the nodes in the networks are the nucleotides or dinucleotides, and the connection between them are estimated by the adjacencies among the nucleotides in the sequence. So in this example, we have a sequence, and here you have AT, you'll be connected to CG because they are close to each other. And the same way, TC, you'll be connected to GG, and CG, you'll be connected to GG, and GG, you'll be connected to GA, and this will form a network that is used to feature extraction. So the main goal of this project is to carry out a comparative study of different types of graphs, like homogeneous, heterogeneous networks, bipartite networks, for DNA sequence modeling. As you can see, different networks generate different features vectors. For example, homogeneous networks provides a feature vector different from a feature vector provided by a bipartite networks. Because the use of different type of complex networks for representation of DNA sequence allows different partners to be found, because the networks have different topology and properties. So this generates different features information. So the main idea is to study different type of graphs for generation of features. So once the sequence have been modeled, it's possible to apply diverse complex network measures to extract the relationship between nucleotides of genomic sequence. So you have the networks, and you can apply different complex network measures to extract partners that will be used to classify the sequence. Representation learning techniques to extract information could be used as well. So we intend to use representation learning techniques as well. And these projects intend to investigate machine learning methods of different classes, like SVM, random forest, neural networks, investigate the performance of different classes of machine learning methods in these features obtained by different types of networks. So you want to study the performance of these methods as well. So here's some reference about some works, and that's it. Thanks so much.
Thank you so much, Fabiana, for your presentation. I will check if we have some questions, otherwise I can go on, but I want to, yeah. So my question is if you have tried some of the machine learning tools that you are mentioning, because you also mentioned the other work that are more based on deep learning models, for instance, that are helpful for avoiding this feature extraction step. So if you have assessed how much you can gain by using traditional machine learning methods with this previous step of feature extraction in comparison with using deep learning methods that can be fed only with the raw sequence.
Mm-hmm. Okay. This project is in the beginning, so I intend to start trying to modeling the sequence as a heterogeneous network, like bipartite networks, and then compare if the method that the works that used homogeneous networks, and then compare with works that use deep learning methods to extract these features. So I intend to start with complex network and complex network measures to extract the features, and compare with features extract with deep learning methods. But it's in the beginning, but I pretend to do it.
Yeah, I think it is very important, and maybe you have read it too, but there are several even currently available models that represent the sequence as embeddings, so through using numeric matrices, and they are being used in a lot of applications related to, or in which you need to take information from the DNA sequence, RNA sequence, protein sequence, even more. So I think it is really interesting, and of course, maybe you can even integrate the two different approach or develop something. Yeah, yes. more even for some applications, these embeddings can be useful, but for other ones, the complex networks can be more appropriate. So of course, I think it is a really nice work, and you have a lot of works and methods and tools for exploring. So
yeah, exciting. Thank you.
Good luck with this.
Hi everyone. My name is Estefania Peralta, and I'm a PhD student in the laboratory of bioactive compound Research and Development from La Plata, Argentina. Today, I am presenting our work entitled Virtual Screening Applied to Search for New Antiepileptic Drugs Active in the PTC Killing Model. Well, the pathology studied in this work is epilepsy. That is a chronic disorder of the central nervous system, one of the most common neurological disorders. Seizures occurs in response to excessive electrical discharges in neuronal cells located in different parts of the brain. Depending on where these excessive discharges begin and how they spread in the brain, will give rise to different types of seizures. From very brief absence give rise to different types of seizures, from or mild muscle contraction to prolonged and severe involuntary movements. Its frequency can also vary from one crisis a year to several a day in extreme cases. Epilepsy affects around 50 million people worldwide, and 80% of patients lives in low to middle income countries. Despite the fact that more than 20 new drugs have been introduced to the market in the last year, around 30% of the patients remain refractory to the available treatment and fail to control these seizures. So for this reason, the constant discovery and development of new drugs are important. Although this is a complex challenge that involves several stages, from identify the biological activity of a molecule to conduct clinical studies that could guarantee safety and efficacy. The introduction of a new drug to the clinical studies should lead to an improvement in pharmacokinetic or minimization of the probability of adverse effects appearance. And it may produce marked changes compared to the already available therapies. There are two main approaches in the discovery of the new drugs. So, those based on molecular targets, for example, ionic channel, and those based on phenotypes, for example, specific cells or animal model. In this work, we will use a phenotypic approach because it is the most appropriate to use in the study of pathology of the central nervous system. As these pathology are usually associated with alterations that involve various molecular targets and alteration in the concentration of endogenous ligands. Well, one of the computational methodology for the identification of new drugs is virtual screening, which is a set of computational techniques that allow exploring the biological activity of chemical compounds that are stored in chemical libraries or digital libraries of chemical structures. This methodology has bioethical and economic advantages because the number of compounds to be tested is reduced, and it allows repositioning. So repositioning is a strategy that focuses on finding a new clinical indication for drugs already approved or at least investigated for other pathology. So repositioning is computational approaches to the search for second clinical indications can be included in 3 groups. First group is bioinformatics methodology. The second group is methodology based on clinical analysis. And the last one, cheminformatics methodology, which is the one we use. In this work, computational models were developed by machine learning to be used in virtual screening in order to identify new drugs with promising antiepileptic activity in the phenotypic model. With this in mind, we performed a literature search for compounds previously tested against this model in mice. The compounds were standardized and the duplicate we removed. So finally, we found 176 compounds that were classified as active compounds and inactive compounds, according to different performance in the PTC killing mirroring model. When we performed a clustering technique to representatively sum the dataset to obtain training and test set. The IRAPCA clustering is based on an interactive combination of feature bagging, dimensionality reduction through principal components analysis, and the K-means algorithm. In this manner, we obtained a balanced training set and 2 validation sets. Validation set 1 was used to evaluate the performance of the individual models in a retrospective virtual screen and to obtain a model ensemble. It was also used to decide which individual models would be combined and how. The second validation set was used to validate the chosen model ensemble using in-house Python routine combining feature bagging, forward stepwise feature selection, and confirmation independent molecular descriptor, we generated 92000 QSAR model. We proceeded to perform an ensemble of linear models using the minimum operator. The ensembles of the top 4 models were calculated and a graph with the number of models versus UseRock and versus Bedrock was plotted. Then one concern when performing virtual screening campaign is the prediction of the probability of conforming an in silico hit when submitted to experimental confirmation. This probability is called positive predicted value, and it is variation with the cutoff can be viewed graphically in a 3D plot display. The interplay between the PPV, the selectivity specificity ratio, and the yield of actives in the screen chemical library. We chosen a strict cutoff score where we would select fewer compounds, but with a higher chance of finding an actual hit. The table displays the enrichment metrics calculated for validation set 2 upon screening with the model and summary, and showing very good early enrichment capacity. So finally, the ensemble of 4 models was applied to a prospective virtual screening of trackband dataset. From the total compounds, 523 passed the strict cutoff score selected previously by the PPB surface. From those, only 371 belong to the applicability domain. We selected 45 compounds approved according to the pharmacokinetics characteristics and the accessibility. Compounds from drug repurposing database could be purchased and evaluated in the PTC killing mutant model. So the epilepsy model used to evaluate possible hits is the PTC killing model, in which progressive changes are produced by repeated application of subconvulsive stimulus. In your case, a chemical stimulus is used, which leads to the so-called Fali-Kindled state that is characterized by the appearance of crisis in response to each stimulus. Pentylentetrazole is a non-competitive antagonist that acts at the picrotroxin binding site of GABA receptors. The endogenous ligand GABA binds to this receptor, which is the main inhibitor neurotransmitter of the central nervous system. This model was chosen because it is relevant to neuroplasticity that simulates epileptogenesis and disease progression. So in conclusion, we generated a computational ligand-based model ensemble associated with very good enrichment metrics in the retrospective screens. The ensemble is able to identify new drugs with promising antiepileptics activity in the phenotypic model. After a prospective virtual screen, in silico hits were selected, which are to be confirmed in vivo in the new feature. Well, I would like to thank the institution that finance and make this research possible. I want to thank you for letting me be part of this conference. Thank you very much.
So let's move on to the next speaker. Netmi, if you want to share the screen. Yeah, you are there. Okay, you are there. Hello. So let's give a warm introduction to Netmi. We can sing. I'm sorry if this pronunciation is not perfect. She's from the University of Moratuwa from Sri Lanka. So glad to have you here. Netmi will talk about anomaly detection in image streams with explainable artificial intelligence. So thanks, Netmi, for coming, and the stage is yours. At 14 minutes, I will tell you that you're on time.
Yes, thank you. Hello, everyone. Thanks for giving this opportunity to share my work. My name is Netmi Vijayasinghe, and I'll be presenting on the topic anomaly detection in image streams with explainable AI. In today's world, there's a large availability of high-quality image streams that can be used for various research purposes, including anomaly detection, through which we can provide real-time support in applications such as deforestation, volcanic eruptions, and flood situations as depicted in shifts here. These are also the datasets we have used. The previous slide contained three real-world applications that our solution can be applied to. But not only in the Amazon, deforestation is also one of the most serious environmental issues in Sri Lanka, with an annual average rate of 1.14%. Not only in the mentioned applications, but such system is also useful in identifying habitat degradations, improper urban development, and medical deformities and distortions. This is where we see the need of a system which can identify such situations in early stages so we can take precautions beforehand. Now let us look at the novelty of our system. We have mainly identified 5 research gaps that we can address in currently existing methods. The first is that the existing methods treat anomaly detection problem as a supervised learning problem. This approach works well with the known anomalies but fails to detect new ones which are not present in the training set. Hence, the generalizability of existing methods is low. As a solution to this, we proposed a novel anomaly detection framework where we treat anomaly detection problem as a one-class classification. The next research gap we have identified is that most of the existing methods ignore the interdependency of the inherent order of the images. We integrated computer vision and time series forecasting to capture the interdependency between the images during the model building process. The third research gap is that most of the existing anomaly detection methods use manual thresholds and unrealistic assumptions. We propose a novel approach to calculate a data-driven anomalous threshold using extreme value theorem. The next research gap we decided to address is that the existing deep learning-based anomaly detection methods solely focus on the classification task, which leads to the lack of explainability of the model. This reduces the human trust on the system. In our proposed framework, we integrated explainable AI along with computer vision and time series forecasting to detect anomalies in image streams. The last research gap we have addressed is the class imbalance problem. To address the class imbalance, some researchers have used different approaches like upsampling, downsampling, and data augmentation, etc., which have their own limitations. So in our proposed framework, we treat anomaly detection problem as a one-class classification problem and build the model for the typical behavior. The aim of this research is to develop a novel framework that detects and interprets anomalies in image streams using computer vision, time series forecasting, extreme value theory, and the explainable AI. There are mainly 3 objectives of this research. The first one is to define a pool of features that encapsulate the signal information content in image streams using computer vision. Second is to propose a novel framework to detect anomalies in image streams in different application domains using time series forecasting and extreme value theory based anomalous threshold. And the last one is to implement a suitable explainable model for anomaly detection in image streams. I'll explain the methodology we followed in the next few slides. In our research, we have defined an anomaly as an observation that is very unlikely given the forecast distribution. Our proposed framework is mainly based on 2 assumptions. The first is we expect that an anomaly shows significant deviation from the typical behavior of a given system. The second assumption is that a representative data set of the system's typical behavior is available to define a model for the typical behavior of the image streams generated by a given system. Shown here is the proposed methodology, which consists of 2 machine learning and deep learning based anomaly detection modules and explainable AI model. The second anomaly detection, the 2 anomaly detection frameworks are subject to comparative analysis in the research, and explainable AI module ensures that the increase in explainability of the black box models. This is the overview of the machine learning module for the anomaly detection. It has an image processing, a feature extraction, principal component analysis, time series forecasting, and classification components. The first component, image pre-processing, takes an image series dataset as an input and improves its quality before feature extraction. The images are cropped, smooth using the Gaussian filter, and contrast stretched. Next, the image dataset is used to extract features, and 4 different types of features are extracted, including first-order statistical, Gabor wavelet features, edge-detected features, and GLCM features. A total of 87 features were extracted by this component. Next, the large feature set is dimensionally reduced using principal component analysis to obtain a univariate dataset, which is then split into training and test sets. The training set is used to build an ARIMA-based time series forecasting model. This is the overview diagram for the proposed framework for the deep learning-based anomaly detection module. The process of this module is very similar to the conventional machine learning module. In the pre-processing stage, we use 3 techniques: image cropping, resizing, and normalizing. Afterwards, we use pre-trained convolution neural network architectures to extract features from the image streams. Here we compare the performances of 5 pre-trained CNN architectures, namely VG16, ResNet 50, Inception V3, Exception, and DenseNet 121 to choose the best model as the backbone of the feature extraction model. To extract features from the CNN, we remove the top fully connected layer from each and add a global max pooling 2D layer to get a single feature vector for each image in the dataset. To obtain a univariate feature series, then we get the average of feature vector for each image, and it is then split into training and test sets. LSTM-based time series forecasting model is built using the training set. Following the ARIMA model of the machine learning module and the LSTM of the deep learning model, we obtain the residual and the forecasted error series. The residual series is used to calculate the anomalous threshold, and then we classify the data points in the forecasted series as an anomalous behavior if greater than the calculated threshold value, else as a typical behavior. A lot of existing threshold calculation methods are based on unrealistic assumptions. The infamous box plot-based threshold calculation assumes that the data set will conform to normal distribution, which will not be guaranteed all the time. As a solution, we propose extreme value theory-based anomaly detection calculation. As anomalies have a low probability of happening, extreme values can be considered to represent anomalies. Therefore, in this technique, a child distribution for generalized extreme values is plotted, and using that distribution, threshold calculation is conducted. The above diagram summarizes the steps of this calculation. Moving on to the explainable AI module. Since deep learning techniques act as a black box model, leaving the understandability and the trustworthiness at stake, explainable AI provides explainability and interpretability. As this is fairly new area in AI, it is important to conduct a proper survey to understand the domain. In this diagram, we have gathered some important terminology, concepts, taxonomies, overall challenges, and proper methodologies. The yellow colored areas are what have been adapted to our study, and the taxonomy given here is used as a basis of our study. The overall challenges that we chose to solve are the problem of adapting explainable AI to real-world scenarios and the trade-off of accuracy versus explainability. For that, we have chosen model agnostic explanation in the local scope and the CNN feature extractors as a black box model for which we provide the explanations. As for the XAI techniques, LIME and SHAP have been selected. SHAP and LIME both give model agnostic post-hoc explanation, hence the accuracy of the original model is not compromised. Both methods aim to identify the importance of each feature toward the prediction of the black box model. LIME and SHAP both decompose the image into superpixels and create a data set by turning on and off the pixel values and getting the prediction from the original black box model. After that, SHAP and LIME follows different approaches in calculating feature relevance. And as in this slide, SHAP takes various combinations of the feature set and calculate the marginal contribution based on game theory. Finally, considering all the marginal contributions of a feature towards a prediction, the total contribution is obtained using SHAP calculation. On the other hand, LIME uses an inherently interpretable local surrogate model to approximate the feature importance. LIME can only give whether a specific feature is contributing to their prediction or not, whereas SHAP can give you the degree of the contribution as well. Though the existing Lime and SHAP methods give reliable explanations, they cannot be applied directly to combination of black box models. Thus, in order to approximate the explanations from the feature extractors, we have constructed 3 proxy CNN classifiers providing the same grounds as the deep learning anomaly detection model. As shown in the diagram, the classification outputs obtained from the CNN classifiers are then separately gone under SHAP and Lime explainers, and each method provides the feature maps with relevant feature scores indicating the feature contribution. This way, we approximate the explanations in terms of feature importance of each CNN model used, as feature extraction lays the foundation for the anomaly detection. Now let's move on to the evaluation and the results of the proposed framework. The results we obtained is summarized on the screen. When comparing the results of the machine learning based model with the deep learning based model, we observe that the machine learning model with an accuracy of 98.3% outperformed the deep learning based model for the small dataset, and for the large dataset, the deep learning based model clearly outperformed the machine learning based model. Another important observation we made from the results we obtained is that our proposed explainable value theory-based threshold calculation method outperformed the box plot-based threshold calculation techniques results in each situation. When we compare the time taken for feature extraction using both the machine learning and deep learning models, we observe that the CNN-based deep learning models took much less time to extract features compared to the machine learning model for both datasets. And also, to prove the generalizability of the frameworks, this table displays the results obtained by the machine learning and deep learning frameworks for an additional dataset that depicts an undersea volcanic eruption. It can be seen that both frameworks provide good results for both datasets, and this generalizability adds to the novelty of the research as was mentioned previously. To give local explanations, we have chosen 100, 1,900 images from the normal class and 2,250th and 2,450th images from the anomaly class to evaluate the slow changes of the normal class as well as the rapid changes once the anomaly is detected. In Lime explanations, the green spots are the superpixels that has contributed to the final prediction in a positive manner, whereas the red spots are what have been negatively influenced. As we can see, ResNet 50 and Inception V3 have identified the most appropriate regions as major contributing areas when identifying an image has an anomaly or not, compared to the VGC16 model. In SHAP explanations feature maps, the variance of pink and blue spotted areas indicate respectively the positive and negative contributions towards the prediction. The higher the intensity of the pink or blue color, more the negative or positive contribution is. Aligning with the Lime predictions, SHAP prediction also indicates that ResNet 50 and Inception V3 have correctly identified the features for each prediction. With that, let's move on to the further work. For further work, the machine learning module can support a more optimized feature extraction and selection process. Deep learning module can be further optimized to accommodate smaller datasets. Moreover, there are a lot of good hyperparameter tuning methods. Since our focus is a little bit different, we left it for the future work. For the explainable AI, a proper evaluation method for the explainable AI component can be introduced, because with the novelty of the domain, we are short of suitable evaluation techniques for explainable AI. I'd like to acknowledge the Retina Research and Innovation Project funded by UNESCO and the International Development Research Center Ottawa for supporting this work. Finally, meet our team. Our team consists of 3 members, myself, Netmi Vijayasinghe, Netmi Sellehewa, and Rashmi Perera from the University of Morato, Sri Lanka, and we are supervised by the Dr. Priyanka Thalagala. Thank you. And I'm open to questions.
Thanks a lot, Nedmi. Amazing presentation. I think we are just understanding, giving interpretation to all the information that you gave us. We have plenty of time for questions. You were perfect in time, so questions are open for the audience. I don't see in the chat maybe some of the
Hi, hi, Nesme. It was a really nice presentation. I really appreciate the level of the detail that you showed to us. It was an impressive work. So congratulations to you and to your group, a full group of women. So congratulations for that. Yeah, I was a bit interested in what is your thinking about, because you showed us that you have differences in terms of performance between the machine learning models and the deep learning models when you are considering smaller data sets or bigger data sets. What do you think about that? I mean, do you think that it is better to have the possibility to count with more data for being able to train and fit a deep learning model? I mean, the time that you need to invest and the resources that you need to invest, do you think that are paying the price that you should pay for having more data? Or what is your feeling on that? And I was also interested in to know if you want to tell us a bit more about the hyperparameter tuning process, how, if you are exploring deeply it, or if you think that you even can improve them for even for achieving better results for this setting with the where you are having smaller data sets.
Yeah, for the hyperparameter tuning question, we have not explored that part yet because it was kind of out of our scope. But we use pre-trained CNN architectures in our deep learning. But if we use like pre-trained CNN architectures and LSTM single layer bidirectional LSTM model in our deep learning based framework. But if we used good hyperparameter tuning methods existing, there are a lot of good techniques existing currently. So if we use those, we can like improve the quality of the and the performances of the deep learning based anomaly detection model for sure. And I don't remember the first question you asked. I'm sorry.
Yeah, it was more, what do you think about this balance between requiring more data but having deep learning models that are more accurate or more precise, or being able to work with a smaller data set with machine learning models that are simpler, that maybe will not require this explainable module, but that could have a lower accuracy, you know?
Yeah, so according to the results we got, what we identified is that machine learning model like performs very well for the smaller data set, but it has like very not that good performance for the large data sets. But deep learning model works well with the large data sets and have a comparatively less but a good performance with the large data sets. So when we compare those two models, I think deep learning model has a better performance compared to the machine learning model in both small and large data sets if we like took as a whole. So I think like if we like do more testing, and if we like do training on more data sets, like we can like increase and improve the performance of the deep learning model to achieve like better results. And also from the time complexity point of view, machine learning takes a lot of time to train the model and to do the forecasting. But in terms of that, deep learning model is very quick, and so we can like apply it to the real world applications very easily.
Oh, congratulations. Thank you so much for your
Thanks
answer.
a lot also for this answer. We have two questions more in the chat. One is what other use or what other use or area could your model be used for? Okay.
Sorry, I didn't get your question.
In which
Shall
other?
I answer?
Yeah.
Shall I answer them in the chat?
Yeah, no, you can answer now. We have some time.
Okay.
Are you,
Okay, how do you manage the images with different resolution level?
Yeah.
So in both machine learning and deep learning models, before like extracting features, we reshape the models to the specific resolution. So that is done in the pre-processing stage in both models. Then for the second question, what are the areas could your model be used for? So as I mentioned in my second slide, except for the deforestation, we can use this in identifying like medical deformities and distortions, and also we can use it to identify any like loss of land cover due to like improvements in agriculture and so on. And also we can use it for identify improper urban planning and stuff. So there are a lot of applications that we can use these frameworks in.
Okay, thanks a lot, Nedmi. We are moving on to the next speaker.
Thank you. Thanks for lot. Thank you, Laura, for this opportunity.
Yeah.
Okay, so good afternoon to everyone. I am Mariana Sofia Candamil, and I am going to talk about calibration and implementation of a predictive computational harvest model for cacao quality improvement. This is the formulation and also preliminary results of my master project. So also I would like to introduce my director, Dr. Jorge William Arboleda, and my co-director, Dr. Angela Camargo. Okay, currently this project is associated with a program called Biofactories, an opportunity for bioeconomics development for Caldas, Colombia through biotechnology. This program involves many sub-projects and techniques to develop bioproducts according to the potential to the species. So this project is only a stage to achieve this goal. Okay. The understanding and responsible use of biodiversity is one of the most important challenges that is currently facing Colombia. The bioeconomy is used to generate an alternative growth path which takes into account the results of scientific research on biodiversity opportunities. Specifically, one of the most important crops in the country is the Oroma cacao, which is a tropical crop cultivated in America for thousands of years. Depending on the pot and ovoid fruit, its mature state can be appreciated with the colors that could be green or red, and also the texture smooth or rough, depending on its genotype. In 2016, an initiative called Cacao for Peace was implemented. Its main objective is to improve cacao value chain in Colombia. That is to take advantage of the fast increase in global demand of this product, presenting an opportunity in the post-conflict context and also for Colombia farming families. The process of cacao includes the seeding, harvest, and post-harvest. Depending on the process, in each step, cacao can be categorized as high or poor quality. Although Colombia cacao has a high potential to compete massively worldwide for its fine flavor and also aroma, the lack of experts and also the lack of emerging technologies focused on its timely production makes the process less than optimal and therefore produces crop yields. Currently, most of the process in this field continue to be empirical. For example, in the harvesting phase, cocoa farmers use morphological parameters such as color, texture, and size to define whether the fruit can be harvested, which is not very efficient since it depends on the stage of the mature, and aspects such as light and texture makes this technique subjective and conditioned to the time and angle at which they observe the fruit. Specifically, harvesting of cocoa generates a decrease in, or ultimate harvesting of cocoa generates a decrease in antioxidants, minimizing the organoleptic characteristics which directly influence flavor, color, and also odor. Similarly, there is an increase in the presence of soluble oxalates, which can have negative repercussion on the organisms, since they contribute to the generation of kidney stones. In addition, crop yields are also affected by climate changes. As such, variability provides an ideal environment for spread the phytopathogens. Therefore, it is essential to implement an user-friendly, also a portable and easy connectivity tools for timely harvesting. Okay, current research has focused on the use of emerging technologies such as artificial intelligence to make a prediction about harvest time. Specifically, machine learning is defined as the use of fine-tuned computational algorithms based on previous experience to generate statistical conclusions about the data. Also to create specific predictions on and also classifications. Their main function is to calibrate and fit the parameters to optimize performance on training data and subsequent the input data. In particular, the use of machine learning models usually requires coding expertise for data processing, training, validation, and testing the data set. In addition, there are many parameters such as the number of iterations, the learning rate, the regularization that represent a great challenge to find exactly the optimal values to generate complex models that properly fit the data behavior. Okay, currently there are several models for the prediction of cocoa harvest. Among these is the one developed by a project entitled CocoLat, which was carried out by BIOS, Hands and Crops, National Institute of Agricultural Botany, NIAB, and the National Federation of Cocoa Growers of Colombia, FEDECACAO. They worked with agroclimatic variables and they developed an app to help the harvest. However, among the results obtained, it was identified that it is necessary to carry out a validation, a verification of the model, and significant impact in the field work with cocoa farmers. Hence, the importance of expanding the existing models by using new data, integrated various models, training strategies, and also take into account other variables like new climatic and morphological data that contribute to an improvement in the implementation and use of this tool. For this reason, the objective of the current project is to implement a computational model for predicting cocoa crop harvest through the use of technological tools that favor sustainable production in the value chain. The methodology of this project includes an identification of the computational model that was made by CocoLat in 2020 and 2021, and also obtaining agroclimatic variables in the site. This includes temperature, wind speed and direction, rain gauge, and also solar radiation. After that, we will continue obtaining more information to the dataset according to each month of harvest, with the objective of identifying when the cacao agricultor has to harvest the fruit. And finally, we will contribute to the calibration of computational model. In this stage, we will create experiments using conventional machine learning like KNN and logistic regression, but also use techniques like deep learning, especially talking about vision transformer, considering the big potential to classify images. And finally, explore auto-machine learning techniques, which can be used to entry to another machine learning models. Okay, here you can observe several preliminary results. We start using images analysis of only 36 images. Each sample was photographed, obtaining 6 photographs for each month of both clones. That in this case, we use ICS 95 and CCN 51 that are both very important here in Colombia. Then we delimited the border of the images and made a colorimetry analysis for each month. The values of red, green, and blue were extracted, and we did an average for each of the colors in each month. It was also measured the texture of each sample considering gray matrix option that is in Python, detecting the dissimilarity, correlation, homogeneity, and energy. In particular, we talked about the dissimilarity gives the information about the distance that we have between two pixels to know how much variance exists exists with respect to the other. For that reason, the average of these parameters was carried out, taking into account all the samples for each month. In the mature period, some index related to morphological features show difference between cacao fruits per analyzed clone, according to the generalization of the profiles of color and texture. And when we talked about CCN clone 51, we noticed that it presents more content of red than ICL's 95 clone. So this could be used as a differential pattern to identify the type of clone. According to the similarity graph, it is appreciated more variation of CCN clone. We can notice here in the graph. This variation is all over the month. The appearance and texture index could be related to changes also in its internal components and internal molecules that it is expressing in a determinate, in a specific time. Okay, for future work, in this moment, the team is focused on collecting more information for each month of harvest for calibrate and prove the model. It is also important to highlight that to prove the quality of cacao, we will also develop a metabolomic analysis, which will help us to understand the specific metabolites associated with quality and then validate the implementation model. This type of research contributes greatly to the economic development of the country, like Colombia, whose main source is income is agriculture. Thus, it is important to identify strategies to optimize the process and to improve the economy of agricultural sector. So here are the references of the work, and thanks very much for your attention, and thanks for all the committee for the invitation.
Amazing talk. We are plenty of time, so also, yeah, and it's nice to see a history of the cacao. No, it's something really interesting apart from the amazing work that you did as a data scientist. So I cannot see any question. If someone from the audience. For Mariana.
Sorry, I can't do the question. Sorry. Thank you, Maria, for your presentation, and thank you for being here. It is a really nice work, and my question is more related to maybe future work, because I agree with you that the development of this kind of tools is very important for the economical development of countries like Colombia and any other countries of Latin America and South America. And my question is if you have a plan about how do you think to release once you have the model fitted, ready for being used more, I mean, outside of the research or an academic environment, right? And in the real life, if you have a plan about how do you, how are you providing the model for its use?
Thank you for the question, Gabriela. Yes, we have been thinking a lot about this, because in this moment, the cacao agricultors, they don't have, for example, smartphones to get an app and to just take a picture, and then to that the application can detect exactly what cacao can they harvest. So we are planning to make a new association with, for example, Fede Cacao, that they and other associations like ASOPROEL here in Colombia, so they can provide that kind of tools to the cacao agricultors, and they can implement this type of researchers directly to the field, because it's also very important that to complement the researcher and investigation with the sector, and can be focus always to improve the quality of life, in this case for the agricultures. I don't know if I answered.
Yeah,
Okay.
yeah, sure. And I agree with you. And I think that this experience will also provide you some feedback, and you then can improve maybe your requirements or the data that you are using on your model and this kind of thing. So, yeah,
Exactly.
I think it's a really nice solution.
Thank you, Gabriela.
Congratulations and good luck with it.
Thank you.
We have another question from the chat, and it's related because I think it's our region. No, the Leda said that in Mexico they have also three principal varieties of cacao. If it is possible to use this model or this method in their culture.
I believe that yes, that it is possible to use these methods and extrapolate the use or its use in Mexico. But I also think that the variety of each cacao is it exists a variety or an amazing variety of cacao. So probably I am not sure if we can use exactly the same model also in Mexico, but I am surely believe that we can use like the same, and maybe with transfer learning use the same model, but with new data to calibrate again and extrapolate exactly the same method to Mexico.
Okay, thanks a lot for being here. Amazing, and also I like the concept of cacao for peace, no? So
exactly,
we are like this idea. Thanks a lot.